Hey Cardionerds, my name is Deepak Atri. I'm a cardiovascular medicine fellow at Brigham and Women's Hospital in Boston. I want to discuss with you a topic that is less cardio and more nerd today. It's the life cycle of SARS-CoV-2, which is the virus that causes COVID-19. We'll then use the framework of the viral life cycle to inform a discussion about therapeutic development. And just one caveat, I'm not a virologist, so if you have some expertise in this area, this talk may be oversimplified for you. And after I finish, my good friend and co-fellow Victor will balance the basic science discussion with an appraisal of the clinical data we've acquired about COVID-19 in these early months. And we hope this is useful and informative for you. So some caveats before we begin. These data are rapidly evolving, as you all know, and so this presentation cannot be exhaustive. It may indeed be obsolete by the time that it comes to you, and where the data are speculative, we have tried to be selective and tell you where the data are speculative. So some objectives for the portion of my talk will be to discuss the coronavirus life cycle and the methods by which a novel virus was linked to the new disease state, to discuss the importance of Ace2, which is the one cardio connection here, of angiotensin-converting enzyme 2 to SARS-CoV-2, and to discuss the molecular basis for emerging therapeutics. So some basic virology to serve as a foundation of today's talk. Viruses can be thought of as vectors for gene transfer. And so they contain nucleic acid, which is either DNA or RNA, encased in an envelope of protein with or without a lipid bilayer. Viruses themselves do not perform any form of energy transduction or metabolism, and are reliant entirely on cellular machinery for replication. They can only gain access to cellular machinery after they've entered cells, and so every individual virus has a receptor on the cell type that it targets. Coronaviruses are a family of enveloped, single-strand, positive-sense RNA viruses, and their genome is generally composed of 30 kilobases. Their membrane, which they have that's studded with protein, is necessary for entry into target cells. And they're called coronaviruses because of this characteristic appearance on transmission electron micrography, as you can see in the figure on the left, where you see the dark halo surrounding the virus. And they are known to cause a variety of diseases in birds and mammals. The 30 kilobase genome of coronaviruses is thought of in two portions. The first portion encodes the non-structural proteins. Together these are called a replicase, which are co-transcribed and co-translated into one 800 kilodalton protein, and then cleaved initially first by endogenous protease and subsequently by the viral proteases themselves, which are the main coronavirus protease or the papain-like protease. Another key protein encoded by the non-structural proteins, or the replicase, is the RNA-dependent RNA polymerase, which is responsible for viral genome replication. Other key proteins of the non-structural proteins are responsible for hijacking and repurposing the endogenous functions of the cell to the aim of viral replication. Classically, coronaviruses have four structural proteins, which are the spike, membrane, envelope, and nucleocapsid proteins. The spike protein is responsible for binding to the target cell. The membrane and envelope proteins are membrane proteins in the viral membrane, which are responsible for the skeleton formation and membrane fusion. Membrane fusion is a necessary step for infection. And then finally, the nucleocapsid protein binds the RNA genome and stabilizes its interaction with the membrane. Coronaviruses bind cells through three classic receptors in mammals and birds. These include aminopeptidase N, dipeptidylpeptidase 4, DPP4, and angiotensin-converting enzyme 2, or ACE2. Importantly, ACE2 is the receptor for SARS-CoV-2, SARS-CoV-1, as well as another common cold virus, NL63. If you notice, each of these three viral receptors are actually proteases, but it's unclear why each of these is a protease, as cell fusion and infection can occur without their enzymatic activity. However, the enzymatic activity of a protein TMPRSS2, which is also a membrane-bound protease, is necessary for infection. We'll come back to the importance of this protein later in the talk. Once the virus has gained access to the cell after binding and membrane fusion, there are several key steps before the virus can be reproduced. These include translation of the viral RNA and cleavage of the subsequent protein products into the replicase and the structural proteins, the amplification of the viral RNA itself by copying through the RNA-dependent RNA polymerase, and it's during this step that recombination and mutation may occur, and then assembly of the viral particles themselves on the endoplasmic reticulum membrane, where the RNA and the proteins meet, and finally require the use of the membrane in order to form a viral particle and subsequent export from the cell. This figure was developed for SARS-CoV-1, but is likely applicable to the life cycle of SARS-CoV-2, and it shows the key steps of the viral life cycle. These are membrane binding, internalization and membrane fusion with the use of TMPRSS2 as I have annotated, genome entry into the cell, whereupon viral proteins are translated and the viral genome is retranscribed, and then finally assembly on the membranes of the endoplasmic reticulum, followed by export from the cell. These steps might be thought of as links in the chain of a viral life cycle, and the virus needs every link in order to replicate. And so if we might drug any one of these steps and break a link in the chain, we might be able to break the viral life cycle and effectively reduce viral infection. And so now we will turn our attention to how an initially idiopathic pneumonia complicated by ARDS was linked to a coronavirus that we now know as SARS-CoV-2. This paper was published in the New England Journal of Medicine at the end of January. Essentially, multiple patients in Wuhan were suffering from ARDS of unclear origin, and given experience with coronavirus in this part of the world, bronchoalveolar lavage specimens were added to a viral culture system, and cytopathic effects were observed. Transmission electron micrography then suggested the presence of a coronavirus, and this was followed up with sequencing efforts, which confirmed the coronaviral nature of the infectious pathogen with its classic genome organization. Parallel efforts were reported in Nature at the beginning of February, which observed comparable sequence homology in RNA and protein. After confirming that the virus in question was a coronavirus, the investigators then sought to identify the receptor employed by the novel coronavirus as a first step to characterize its life cycle. They began with an assessment of angiotensin-converting enzyme 2, which I will call Ace2 for the rest of this talk, and they did so because the original SARS coronavirus also used Ace2. This is a clever experiment performed in HeLa cells, which are a cervical cancer cell line that do not express Ace2. On the far left column, you see HeLa cells with their nuclei stained in blue, and you see that they do not express Ace2 as attempted with a green stain. When virus particles are added, you do not see that there is expression of SARS-CoV-2 N protein inside the cells, as stained in red. This tells us the cells didn't become infected. In the second column, we repeat this experiment after adding mouse Ace2 to the HeLa cells. The addition of mouse Ace2 to the cells and then viral particles also didn't result in expression of viral N protein within the cells. However, when Ace2 from civets, pigs, bats, or humans are added to HeLa cells and viral particles are then added, N protein is expressed. Thus, the cells have become infected. And this is important for two reasons. One, it suggests a mechanism of zoonosis, or the manner by which viruses become transferred between different species to humans, and then it also suggests that wild-type mice might not be useful as a model organism in the study of SARS-CoV-2. And now a word about Ace2, or angiotensin-converting enzyme 2. This is a protein that is less known to cardiologists than its cousin, Ace1. The protein is broadly expressed in the human body with detectable expression in the lungs, namely the type 2 pneumocyte, as well as the heart, the endothelium, and the GI tract, among other tissues. This protein functions as a protease to cleave angiotensin 2, which is a vasoconstrictor, into angiotensin 1,7, which is a vasodilator, and thus Ace2 counteracts the effects of Ace1. The protein is encoded on the X chromosome, and that may account for sex differences in infection, as Victor will discuss later. Finally, the enzyme Ace2 is actually upregulated in expression, at least in mice, by ACE inhibitor as well as ARB use, or angiotensin receptor blocker use, which is why some have hypothesized that it may be harmful and increase predisposition for SARS-CoV-2 infection. And now to turn our attention to the effects of ACE2 in the lung, it seems that ACE2 deficiency is protective in the development of ARDS in animal models of this disease state, as evaluated histologically and by measures of compliance. You can see this in a representation on the right, wherein in the top two panels, wild-type mice and ACE2 deficient mice have saline infused to their lungs before they are fixed for histology. On the bottom right, you see an acid aspiration model of ARDS in ACE2 deficient mice, and the appearance, histologically at least, is more severe than that of the same experiment performed in wild-type mice, suggestive that ACE2 is protective of ARDS. It was further observed that ACE2 levels were lower in wild-type mice when ARDS was induced, and that these effects could be at least partially rescued by the administration of recombinant human ACE2. Using this animal model of ARDS, we again observe that the effects of ACE2 deficiency are balanced by the effects of ACE1 deficiency. We again observe that ACE2 deficiency results in a very severe phenotype of ARDS in the top right panel compared with the top left panel, the wild-type mouse. Then, knockout of one allele of ACE1 partially rescues these effects, whereas a second allele knockout of ACE1 further rescues the effects. This suggests that the effects of SARS-CoV-2 infection appears to result in myocardial injury in its severe forms, and pertinent to this we look back to the effects of ACE2 deficiency on myocardial function. We see that ACE2 deficiency results in a cardiomyopathy and a contractile deficit as characterized in panel B with echocardiographic assessment of ACE2 deficient mice. And to further support the notion that ACE1 deficiency counteracts the effects of ACE2 deficiency, we observe that this contractile deficit is actually rescued on the bottom right by ACE1 deficiency.